All right, it looks like we're ready to begin, so let's get started. So hi again, welcome to today's webinar. Just a quick intro about myself. My name is Yuan Tian. I am the Content Marketing Manager at Biocelgene Boston Corporation. I will be your moderator for today's webinar. So for today, we're really excited to bring you this session focused on target humanized mice for toxicology studies, and we've got some great insights and case studies lined up. With that said, I'm joined today by two amazing speakers, Andrew Stansiper from Apertura Gene Therapy and Jay Zhang from Biocelgene. So I'll start with a quick introduction of both speakers just to give you a bit of background before we hear their stories. So first up, it's my honor to introduce Mr. Andrew Stansiper. So Mr. Stansiper is the acting chief technology officer at Apertura Gene Therapy and also serve as a director and gene therapy program lead of Deerfield Management's discovery and development team. Prior to Deerfield, Mr. Stansiper was a consultant for over 3 years at Duck Horse Consulting Group, where he provided strategic and tactical support for more than 30 cell and gene therapy firms, pharma and investment firms. And previous to Dark Horse, Mr. Stansiper worked at 4D Molecular Therapeutics in several research and process development roles. He holds a BS in chemical engineering with an emphasis on biotechnology and an MBA from the University of California, Berkeley. He also serves on the scientific advisory board for JAX Tomorrow, a foundation working to develop a treatment and ultimately a cure for Apertura syndrome. So thanks for joining us today as a speaker, Mr. Stansiper. We're really glad and honored to have you with us today. And next up, it's my pleasure to introduce Dr. Jay Zhang from Biocelgene. So Jay is a Regional Business Development Director at Biocelgene Boston Corporation. He leads a team of 11 business development managers and oversees our biomass business along the West Coast. His work focuses on drug target humanized mice, gene editing in mice, and preclinical pharmacology services. Before joining Biocelgene, Jay spent 4 years as a postdoc and drug discovery scientist focused on ion channel diseases at a local startup in Missouri. He earned his PhD in bioengineering from the University of Missouri Columbia University, where he trained in electrophysiology and biophysics to study the CFTR chloride channel, which is linked to cystic fibrosis. So welcome, Jay, and thanks for joining us today. All right, now that we've met the speakers, just to quickly go over to this agenda, we will have Jay kick things off by sharing recent insights on the FDA's new policy. And then Andrew will take over to walk us through Apertura's exciting CNS gene therapy approaches and application of humanized mice for their tox studies and more. And finally, G will come back to share more case studies and give us a look at what Biocelgene can offer in this space. All right, with that, just a quick note before we begin. This webinar is being recorded and we will leave time for Q&A at the end. So if you have any questions as we go, feel free to drop your questions into the Q&A box and we will get to them after the presentation. All right, now without further delay, I will hand it over to Jay.
Thank you, Yuan. Hello, everyone from coast to coast and all over the world. Thank you for joining our webinar focusing on drug target humanized mice for IND-Enabling Toxicology studies. My name is Jay Zhang. I'm the Regional BD Director from Biocelgene. Today, we're very honored to have Dr. Stansiper from Apertura Gene Therapy to join our webinar and share their exciting story. So before we jump onto the toxicology topic today, please allow me to use this one slide to quickly and briefly introduce Biocelgene's comprehensive products and services. Biocelgene is one of the world's best gene editing service providers to customize mouse models for our partners. Over the past 17 years, Biocelgene has delivered thousands of custom models to our partners from academia to industry. Meanwhile, to meet the unmet needs of in vivo animal models for drug discovery and development, Biocelgene strives to provide thousands of off-the-shelf drug-target humanized mouse models and cell lines for in vivo studies. And as a CRO, Biocelgene has established a comprehensive preclinical pharmacology service platform to conduct efficacy study, PK/PD study, and toxicology studies, with thousands of successful projects delivered. So in today's webinar, we will focus on how the drug-targeted humanized mice can be used in IND-enabling toxicology studies. We will use 4 sessions to cover the toxicology topic today. So first, let's revisit the importance of toxicology studies in drug discovery and development process. We will summarize the regulatory and industry trends, as well as the scientific rationale of using drug-targeted humanized mice. Second, Dr. Andrew Stansaper will share their exciting story of IND-approved toxicology study with us. Following by Andrew's presentation, will be 3 IND-approved case studies that use drug-targeted humanized mice for toxicology studies in their submissions. So lastly, we would like to provide an overview of Biocelgene's in-house toxicology capability. Now let's begin. So the figure from Dr. Raymond's book, Drug Discovery and Development, illustrated the timing of the main safety assessment studies during general drug discovery and development process. Before the IND submission, we should perform non-GLP toxicology studies in vitro and in vivo, including single and repeat dose range finding studies. Furthermore, we should also perform GLP toxicology studies, including safety pharmacology, genotoxicity, and repeat dose toxicity and recovery studies. Once we enter the clinical phase, we need to perform long-term GLP toxicology studies and reproductive toxicity studies. As we all know, the preclinical toxicology studies are mandatory for IND submission to regulatory bodies such as FDA or EMA. These studies play a critical role in safety assessment to help identify potential adverse effects before first-in-human trials. From these studies, we can determine the starting doses and estimate maximum tolerated dose for clinical trials. And ultimately, the preclinical toxicology studies can help lower the risk of clinical failures and financial losses. For the in vivo toxicology studies, selecting a relevant species is critical. Thus, the regulatory bodies provided several guidelines on selecting the relevant species. According to the ICH I6 in 2021, a relevant species is one in which the best material is pharmacologically active due to the expression of the receptor or an epitope. So by this definition, the conventional animals used for safety evaluation are not relevant species. Traditionally, non-human primates are relevant species if the candidates cross-recognize human and non-human primates. But when the candidates do not cross-recognize human and non-human primates, and no relevant species naturally exists, then the transgenic animals expressing the human receptor should be considered. Then in our case, it means drug-targeted humanized mice. Of course, using mouse surrogates is an alternative strategy, but it has its own list of disadvantages. In 2022, during the COVID-19 pandemic, FDA strongly encouraged the use of drug-targeted humanized mice for toxicology studies, especially for DART studies, due to the disrupted supply chain of non-human primates. The needs and the interest of using drug-targeted humanized mice for toxicology studies increased a lot since then, and now it has a strong momentum. Earlier this year in April, FDA announced to phase out animal testing requirements for monoclonal antibodies and other drugs, especially the use of non-human primates, for 3 major reasons. The first reason is, of course, the ethical issues, strict welfare standards, conservation status of these non-human primates, and public pressure. Scientifically, some safety risks may still go undetected in animals with monoclonal antibody TGN-1412 as an example. This antibody caused a less threatening CRS in human despite appearing safe in preclinical monkey studies. Furthermore, cross-recognition does not mean the affinity can be translated well from monkey to human. The last reason is the high cost and time investment using non-human primates. Monoclonal antibody development programs typically use 144 non-human primates, and the cost goes up to 50,000 per animal. During the phase-out transition, FDA suggested using humanized transgenic animals as one of the new approach methodologies. So the drug-targeted humanized mice can help a lot during the phase-out transition to serve as a great alternative relevant species. To recap, drug-targeted humanized mice are a reliable option when relevant species are limited, as supported by ICH I6 guidelines. Drug-targeted humanized mice serve as an alternative to non-human primates for DART studies as recommended by the FDA. In the effort to reduce animal testing, especially non-human primates, humanized transgenic models are suggested as a new approach methodology by the FDA. What if we compare the drug-targeted humanized mice with non-human primates and surrogate antibodies? The drug-targeted humanized mice are genetically engineered mice in which the specific human genes replace their mouse counterparts. This allows in vivo evaluate drugs against human-specific targets. So when we compare that with using non-human primates, drug-targeted humanized mice are cost-effective and efficient. It will save up to 50% cost with 100% quality guarantee. And also the study takes shorter time and consumes less amount of test articles due to the lower body weight of mice. If we compare with using mouse surrogate antibodies, the drug-targeted humanized mice are more reliable. They have higher relevance for human-specific targets and preserves the target specificity and mechanism of action. That allows the direct evaluation of drug candidates for clinical trial candidates. When we use drug-targeted humanized, when should we use drug-targeted humanized mice for toxicology studies? Here is a similar time flow of the drug discovery and development process. The drug-targeted humanized mice can help start from the beginning of model establishment, followed by in vivo efficacy study, mechanisms of action, PK/PD studies, and toxicology studies. All these data package will be used for IND application. So before the IND application, the preclinical toxicology studies, we can do non-GLP toxicities. In these non-GLP toxicity studies, drug-targeted humanized mice are generally recommended. That enables evaluation of on-target toxicity against human targets and its cost and time effective. And for GLP general toxicity, especially when the non-human primates do not cross-react with the candidates, then the drug-targeted humanized mice are extremely helpful. So in the IND phase, of course, the non-human primates, in the IND phase, in the clinical phase toxicology studies, the drug-targeted humanized mice are also very helpful in the reproductive toxicity studies or long-term studies. So without further delay, I would like to hand my microphone to Dr. Andrew Sapper. Looking forward to his exciting story. I will just stop sharing.
Thank you so much, Jay. It's very kind of you. I'm going to bring up my screen. So give me 1 second here as I click the buttons. So now I believe everyone should be able to see my screen. It's a pleasure to get to be here. I really appreciate the opportunity. As Jay noted, my name is Andrew Steinsapper. I was not smart enough or driven enough to go get a PhD, so not doctor. My wife is, though. She's much smarter than me and much harder working. So my name is Andrew Steinsaper. I am the Acting Chief Technology Officer for Apertura Gene Therapy, as I was noted earlier. I also work at Deerfield Management. Deerfield Management is an investment firm. That's my full-time job. I am the Director and Gene Therapy Program Lead there. I've been there for about 4 years. We'll be talking a bit about Apertura's work and our feedback from FDA as we've developed our programs. A little bit about me, so you know who's talking to you. Here's a picture of me because I'm not really sure if you can see my face right now or not. This is me looking good on a good day versus disheveled as I probably am right now. My background is in chemical engineering, as just noted. I've spent my whole career focusing in cell and gene therapy drug development. I've had the opportunity, though, to work on small molecules and large molecules now as well, usually spanning anywhere from that preclinical, operational, or other set of opportunities. My experience across these has not just been as an innovator, but also supporting folks as a consultant and thinking about those problems, moving them forward. Talking to you today, I'm going to focus on what matters, or at least what matters to me, which is not academic publications, but rather developing drugs and hopefully being able to develop safe drugs that are efficacious, move them through clinical trials, and eventually approval. Oftentimes, now I'll be speaking about gene therapy perspective, oftentimes this can take a very long time, but in gene therapy can go much faster, which I think is another reason why it's important that we start with the end in mind and working with the right programs. So I'll kick into the Apertura presentation now. So Apertura Gene Therapy overall, we're a gene therapy company. We're focusing on developing therapeutic products, but also we have to build tools that allow us to do that. So before we dive into our novel pipeline or talking about our capsids, I think it's important just that you understand how we're solving the problems in gene therapy and the biggest challenges and how we differentiate ourselves from others. So for more than a decade, capsid engineering, which is what we do, has largely relied on brute force approaches and iterative library design and screening in animal models, typically non-human primates, to generate data. So the introduction of machine learning into these efforts over the last couple of years has yielded economies of scale, but historically, capsid engineering still relies on using brute force efforts in what we believe are fundamentally flawed premise, and that is trying to develop something in an animal that is not what you're going for, because the thing we care about, again, is human translation. So our approach instead is differentiated because we start with that end in mind. We want to work with human receptors because we have human-centric efforts of developing drugs for humans, not for mice, not for non-human primates. And that's one of the reasons why I'm here today. We rely heavily on humanized mice so that we have a translatable and specific and selective opportunity when doing this development. This also helps us as we think about that end in mind. It's not just about finding something, but knowing that it's going to work later on. And since we know what it interacts with, how it interacts, and how it moves forward, we believe we can get shorter timelines in running development. And then as a result, we're also making sure that our capsids actually can be functional and manufacturable. All of this work was pioneered by Dr. Ben Deverman at the Broad Institute, who I believe has spoken for Biocytogen in the past, more than a decade of work here. And it's a result of his hard work that these things have moved forward. Ben is the scientific founder of Apertura. Apertura itself has an elite profile of different capsids and portfolio. We're happy to be flexible. My goal here, though, is not to give a sales pitch. Don't worry, Biocytogen team. I'm actually here to talk about our development of programs. So overall, and as a company, I've already given you a little bit of background in Apertura Gene Therapy, but for some folks on here, it might be your first time hearing about gene therapy other than things that you hear in the news. So as an overall, one of the things that we're doing as Apertura and changing things is that we use our proprietary receptor-targeted capsid engineering platform to develop AAV vectors. These are the viruses that can specifically bind, in this case, to human receptors of interest, and thereby increase the likelihood of clinical success in patients because of that translatable link. This mechanism-first approach can both de-risk the transition from animal models to humans, and I think important and critically, reduce the time and cost in preclinical development. Now, with that, the things that you're seeing on the screen right now, this is a mouse brain, and all of those green dots are actually successful viral genomes that have been delivered to different cells. The blue, of course, for those that are in the know is DAPI, that's standing nuclei, and what you see here is the entirety of a mouse brain being suffused with virus, because an IV route of administration gives you great access to the brain. So with this, why do we need something like this? Well, we need something like this because the existing status quo with AAV9, which is what you're seeing on the left, really doesn't do this. There have been approvals and successes in the gene therapy space. That's absolutely true and clear and something that I'm excited by, but we can do so much better. And especially as we move to more complex diseases, we need better abilities to cross the blood-brain barrier. We need to be able to get deep structures that are important for different diseases. We need to think about how we can lower the dose and the amount of virus needed to improve safety opportunities. And we want to be moving to routes of administration like an IV or intravenous route rather than direct injection into the brain or into the CSF. Now, with all of that, we also want to be able to maintain manufacturability. So there are a lot of jobs that we need this little virus to do. And the way that we get these things to move forward is by using the knowledge that we're able to develop from the biocytogen mice. So mentioned it earlier, we use the human transferrin receptor. Our AAV capsids have proteins encoded into them or amino acids encoded into them that allow them to grab onto the human transferrin receptor. For folks that aren't neuroscientists, which I myself am not, I don't have the right letters after my name, but I work with a lot of them. The human transferrin receptor that our lead capsid named TFR1 CAPX specifically binds to is highly expressed on the vasculature of the blood-brain barrier, specifically on the capillaries. And that receptor can shuttle biomolecules through the barrier, through the blood-brain barrier, through a mechanism that's known as receptor-mediated transcytosis. Human transferrin receptor is pretty much the most well-studied receptor in the brain for doing this. And so we didn't invent this idea. Many folks have been thinking about this and trying to do this. And in fact, some people have been successful. So using this receptor, we can deliver our CNS-targeted AAV particles through an intravenous route of administration, which is minimally invasive as compared to others. And importantly, this route of administration scales across species and across mammals, which promotes the translatability when moving from preclinical animal models to humans and predicting dose. This is critical. I think it was noted earlier about the requirements of developing data when presented to the FDA, EMA, and others. And one of those critical ones is that the route of administration you're using in the models that you're using and the exposure that you get is representative. We have the ability to check all those boxes. The human transferrin receptor has been very well studied and validated by other companies. And it's something that we're excited about, that it's not just us that are seeing this work, there's others, including folks with clinical and approved products that have validated this utility of this receptor in shuttling complex biomolecules across the brain. So what does that mean? We have something that can get to the brain. Well, there are a lot of diseases that we believe start with and end with the brain. I have some of those listed here for folks that aren't necessarily well-versed in the rare disease space. Some of these ones down below might be newer ones, but when we think about the brain as a complex organ, there are different parts of the brain that might be associated with certain diseases and others. And as you could see in our previous picture, we're able to get to everywhere in the brain. So that means that with something like this transferrin receptor capsid, TFR1 CAPX, we unlock all of these opportunities to move things forward. Now, I'll talk a little bit about a collaboration at the end, but the important thing that I want to note here is that there's not one single company that can go after every single disease. And we don't think that Apertura is going to be able to do that. And we could use the mice that we have access to through biocytogen, and so could you, in order to go after these. But critically, we're interested in collaborating and making sure other people have the opportunity to do this, because together we can do much more than any one of us can do individually. Now, one of the programs that we developed internally at Apertura was tuberous sclerosis complex 1, or subunit 1, TSC1. We have other programs that we're developing and other ones we're collaborating on, but I'm going to use this as our case study today. So for the folks that aren't familiar with it, tuberous sclerosis complex, or TSC, which I'm going to call it from now on because it's a really long number of syllables and I'm going to mumble it if I don't. So TSC is a rare disorder that affects multiple organ systems, as you can see here. TSC affects approximately 1 in 6,000 live births and is the leading genetic cause of epilepsy and autism. TSC-associated epilepsy generally begins during the first year of life and is associated with neurodevelopmental and cognitive problems. Management of the epilepsy in TSC remains an unmet need, with a large fraction of patients being resistant to pharmacological or surgical treatment, meaning that existing anti-epileptic medicines are not effective for these patients, meaning that they often cycle through 2 or more and still are having seizures. TSC is caused by an autosomal dominant loss of function mutation in either the TSC1 or the TSC2 gene. You can see part of that complex here on the left. It's actually much larger than that, but I don't have room in the whole screen, and this isn't a structural talk. The TSC complex regulates the activity of the mammalian target of rapamycin, or mTOR signaling pathway. Now, it's probably lighting up a lot of alarm bells for folks in this audience because mTOR is everywhere. Loss of function mutations in TSC complex cause mTOR and the downstream pathways to be hyperactive, and this results in abnormal cell growth, proliferation, and epileptogenesis. Now, when we think about this from a gene therapy context and perspective, we believe that AAV gene therapy can address the root cause of TSC. That's because we're targeting the nervous system. We need to drop off information and do it in cells that are not dividing anymore, and we'll be able to retain that capability. So Apertura, as a result, has developed AGTX1111. This is a recombinant AAV using our lead capsid, TFR1 CAPX, that uses the human transferrin receptor in order to cross the BBB. It's designed to broadly distribute across the CNS and deliver a healthy copy of the TSC1 cDNA after IV administration. So the TFR1 capsid is able to efficiently transduce neurons and astrocytes throughout the brain, all the cells in the brain, and it offers an opportunity to see other parts of the body as well, because TSC is something that there is no such thing as an off target. Now, our goal here, though, is to focus on the brain, because we're trying to go for both the epilepsy and then the neuropsychiatric disorder TAND that's associated with it. Now, it's great to have an idea, but the most important thing is, as I noted earlier, getting these things to patients, not just in a clinical trial, but getting a drug approved and making sure that everyone has access to it. So the only way to that point is to work with regulatory bodies. So we always start things at Apertura with the regulatory strategy in mind. So when talking about TSC, our first set of questions are, how are we going to develop this? What evidence do we need to develop? And rather than just trying to come up with things on our own, we instead went to the FDA and asked them a series of questions. We executed what's called an FDA INTERACT meeting. So again, as noted, like at Apertura, we believe that regulatory strategy is everything. It should drive preclinical development because we're not focused on papers, we're focused on developing medicines. So we were interested in engaging the FDA early through the INTERACT meeting in order to obtain feedback on our therapeutic development strategy. We wanted to seek alignment on 4 points critically. So the first was that the epilepsy associated with TSC mutations could be classified as neurodegenerative disorder. That's critical for development questions and what level of evidence you need and how to run trials. We're not going to focus on that today. The second was that we could use the humanized transferrin receptor knock-in mouse model that we have access to because biocytogen, and use that as a definitive species for the evaluation of biodistribution and safety of our drug candidate. And at the same time, that non-human primates were relevant for that evaluation. I'll tell you more about that one later. The third thing that we asked to talk about was establishing and using a surrogate capsid approach to evaluate and assess dose range findings in the disease model. This is exciting for folks that are in the gene therapy space. This part will be very interesting to you. And then last one was asking about a cell-based ELISA and an assay that we could use to develop for a potency assay. We put forward all of these questions and communications to the FDA, and we received positive feedback across the board on all 4 of these questions. For the purposes of today's talk, though, I'm going to focus on questions 2 and 3, talking about the animal species selection questions and the dose range finding, because both of these matter a lot for the use of the biocytogen animal. Now, with that, these are not the only programs that we're working on. So critically, the FDA has provided clear guidance on Apertura's regulatory strategy for preclinical development using the TFR1 CAPX gene therapies as a whole. That's not just for TSC1, but also for our work supporting and collaborating with Broad Institute for their Prion program. The 2 key components of our strategy are exclusively using the humanized transferrin receptor knock-in mouse model as the species for distribution and safety evaluations of our drug product. And that's without requiring any additional non-human primate studies and the use of that surrogate capsid for dose ranging studies. The FDA endorsed this strategy in its feedback on our INTERACT submissions, both for our efforts as well as for the BROADs that we were supporting. That's in 2024 and 2025. And critically, this work being developed by Dr. Valab's efforts at the Broad Institute of Harvard and MIT, which we'll talk a little bit about, but just alluding to, these are both very complex things. We have a gene supplementation approach. They have a much more complex epigenomic editor, but still with this, there's alignment with the FDA on this strategy. And frankly, as Jay was noting earlier, all this has already been spelled out in guidance from the FDA and ICH, and anyone could have figured this out. We happen to, and I want to make sure I share that with you so you don't have to go through all that hard work. So critically, the capsid we have and that we've developed is specific to the human transferrin receptor. So we've characterized this down to the amino acid residues in the human transferrin receptor that are necessary and sufficient for TFR1 CAPX to bind to the human transferrin receptor. Those key residues are not present in any non-clinical species, including non-human primates. So in other words, as we understand it, everything down to the amino acid level, we know why TFR1 CAPX is specific to humans and why non-human primates are pharmacologically relevant. As a result, we know not to use them. And to Jay's point, there's already guidance saying not to do that. I'll note that we conducted non-human primate studies early on at Apertura in order to demonstrate and develop evidence of absence of an outcome. We have both in silico, in vitro, and in vivo data now proving this out. Now, not working with non-human primates offers several advantages. I think some of those were outlined earlier. Now, obviously, there's an acceleration opportunity for timeline to IND allowance. I think it's important to call out the reduced cost of animal studies and manufacturing because that the body weight ratio is noted earlier. And the thing that I think about when running development is that I'm looking for rare events when I'm running safety studies. As a result of looking for rare events, the only way to do that is by increasing my N. Working with mice allows me a higher experimental throughput and certainty from working with that larger number of animals. And as a result, I have improved resource utilization. So with this, the thing I'm going to move on to is like, why do we know that this mouse developed by the team over at Biocytogen is useful? And really what that comes down to is how it's built. So instead of working with those non-human primates, our strategy relies on working with that humanized transferrin mouse. We've been working with, specifically, it's the B-HTFR1 strain from Biocytogen, which expresses a chimeric transferrin receptor containing the human sequence on its extracellular domain and the mouse sequence in the intracellular domains, all under the same regulation as the endogenous mouse transferrin receptor and the same 3' UTR. And what we can see on this Western blot is that the transgenic mice express transferrin receptor at the physiologically relevant levels, similar to the expression seen in the background strain, the C57 black 6Js. Now, therefore, what we see is that this model is going to allow us to accurately look at biodistribution toxicology of TFR1 CAPX gene therapies because it uses the specific receptor. And indeed, what we do see is that we have confirmed enhanced distribution of TFR1 CAPX to the CNS in this mouse strain. I showed you pictures of it getting into the brain earlier versus what we see in AV9. There's also precedent for this approach. In addition to our strategy being aligned with published FDA guidance, other companies, some of them were mentioned earlier, have also leveraged specific transferrin receptor mice for their development efforts. I'll note that the approved product from JCR Pharma has, because it's approved, you can download all their information from PMDA, the Japanese equivalent of the FDA. You can see that they used humanized mice in order to do the development. Denali, which has filed a BLA for their product using transferrin receptor, also published efforts using a human transferrin receptor mouse. Now, with that, we have other examples of companies that are developing therapies, not just AAV, but also enzymes, antibodies, and RNAi therapy using the human transferrin receptor to cross the BBB. Several of these are using transgenic transferrin mice. And one of the things that I think is great here is that Biocytogen has the opportunity and availability of making sure that you can use their mice. We use them heavily. But as you can see, this does cover candidates across a range of modalities and a range of development stages. When it came to translating dose, what we did say, well, we could try to cross the mouse models that we have, and crossing that humanized transferrin mouse with the disease model. And this is probably the most straightforward way of using things in developing a path. But what we didn't want to do here is have to reinvent the wheel, recharacterize a disease model, instead more rapidly move forward to developing therapeutically relevant data. So what we did instead is propose a different approach. I'm not saying that any of this is wrong, but there are opportunities to move more quickly and still retain that ability to understand what is happening and be able to present data to the FDA. What we presented to the FDA and what we have endorsement on is what we call a surrogate capsid approach for an efficient preclinical development. So instead of crossing a disease model with the humanized transferrin model, both Apetura's TSC1 program, the Broad Institute's PRION program, and others are using the surrogate approach. As part of the two separate INTERACT submissions, the FDA has agreed to the use of the surrogate approach. So what is that approach? What it does is we have disease models that are going to be evaluated separately using a capsid that works specifically in mice and uses a mouse receptor. And then after that, use the transferrin mouse and compare distribution outcomes. So this removes the requirement of additional breeding. It allows us to leverage data that's already available or folks have generated in the therapeutically relevant disease model with other surrogate capsids, and establish a platform approach by applying the same strategy to future programs that utilize the same surrogate capsid in preclinical development. So what we do is we use a BBB crossing capsid. In this case, it'd be PHPB, which is used heavily in neuroscience, PHPEB, you can see this actually down below, as well as a capsid called BI28 that works in other mouse models. The capsids are all IV administered. They cross the BBB and utilize the same mode of action, which is receptor-mediated transcytosis. So after they bind to those receptors that are expressed on the BBB, they cross over. As you can see here on the top, this one here is using a transferrin capsid and getting into the brain of a transferrin receptor knock-in mouse. This is in the same transferrin receptor knock-in mouse, but instead using the mouse-specific receptor capsid. In this case, this is PHPB. We're looking at episomes here, so those are actually viral genomes. Then down below, you have TFR-CAPX in a transferrin receptor knock-in mouse, and we're seeing the immunofluorescence of the expressed payload. And then on the right-hand side, you're seeing the same mouse model, but now instead using PHP-EB to cross an immunofluorescence that's there. So what this does is allows us to quickly and efficiently transition and understand how much virus is giving us an outcome rather than needing to recharacterize models and start from scratch. Now, like this capsid correlation approach, this, I know this is a lot that's on here. What we do and what we have on this slide is a demonstration of how preclinical development plans can use this surrogate capsid to establish that outcome. So our goal is to bridge data across studies and establish correlations in gene delivery efficiency between the clinical capsid, TFR1-CAPX, and the surrogate capsid. And in this example, that's PHPB. This is a really complex slide, and I get that, but I'm going to go through the different panels from left to right. So first, on panels A and B, we have our disease mouse. So in this example, it's the TSC1 conditional knockout mouse, and their non-disease counterpart in the same background. So we use a surrogate capsid, in this case, PHPB. We assess gene delivery efficiency as a function of dose and understand the dose response in the gene delivery and disease outcomes. We'll also confirm how the capsid's delivery efficiency compares across the disease and non-disease mice. This allows us to establish a relationship between A and B. Then on panel C, we continue the work with the surrogate capsid, but now in this context, we're using the biocytogen humanized TFR1 knock-in mouse. Using that surrogate capsid, we can assess the gene delivery efficiency as a function of dose, and then confirm the surrogate capsid's delivery efficiency compares across the disease strain and the TFR knock-in mice, and establish the correlation between A, B, and C. On panel D, we're now working with our clinical capsid, TFR1-CAPX, in this context, in the human transferrin knock-in mouse. We do the same thing. We assess the gene delivery efficiency as a function of dose, and now we can compare all of these with A to C, A to B to C to D. With that, then we have our last panel, which is the important one. On panel E, we're working on a dose extrapolation to human patients. The FDA INTERACT submission proposed that the expectation that for TFR1-CAPX and other TFR1 binding AAV capsids, like our antibody evading capsid, TFR-CAPXE, that the viral genomes per kilogram dose in a humanized knock-in mouse will be consistent with the human dose on a viral genomes per kilogram basis. The FDA agreed with the scientific rationale behind this approach. This allows us to confirm the relationship between D and E. So to make these assessments in gene delivery, we'll be using two types of analytical methods. Critically, we're looking at immunofluorescence here, as well as in situ hybridization and ddPCR for bulk analysis to see how many viral genomes are in those cells. Comparison of that gene delivery data across studies in combination with data from IND-Enabling Tox studies will then guide dose selection for future studies. I apologize because my mouse just freaked out. This slide, which follows that one, is a little bit of the math of how we get there. I want to note that like all the numbers on here are for illustrative purposes only. I asked my team to help make a slide like this for me, where the math is easy for me to explain and for others to do in their head, because I work with a lot of nerds that can do some really hard math. So please note all these notes are illustrative purposes only. So what you're seeing here is effectively, how do we go from an idea to developing a therapy? We're going to do critically a couple of studies. The POC study is where we use that surrogate capsid. In that study, we injected mice with PHPB. We found that a dose level of, say, 2E13 viral genomes per kilogram gave us that robust improvement. That's our minimum effective dose. I think, like Jay was noting, these things, or we'll be talking about these sorts of regulatory ideas later. In the interim, in between that, we run a capsid comparison reporter study. This is where we're using that transferrin receptor knock-in mouse. And we're using both our surrogate capsid as well as TFR1 CapX. And here we're finding the relationship and how much better TFR1 CapX is versus that surrogate capsid. In this case, we see that there's a 5-fold improvement in TFR1 CapX's ability to drop out viral genomes. Then what we're going to do is, using that information, we also run a safety study or a tox study using the drug candidate, which is TFR1 CapX, the therapeutic payload, and because of our feedback from the FDA, that transferrin knock-in mouse. In that, we see that we have well-tolerated outcomes across all dose levels, up to 1E14 VG per kg, and then at 5E14 VG per kg, we had some treatment-related tox findings. So our NOAEL, or no observable adverse effect level, is at that 1E14, which gives us our upper end. Now we can do all the math that's on the bottom of that screen, where we have our minimum effective dose. We convert that from PHPB over to the surrogate capsid over to our therapeutic capsid, TFR1 CapX, and we have a range from 4E12 VG per kg to 1E14 VG per kg as an example. Now, with all this, what I hope that it highlights is that this is actually straightforward. It is outlined in the guidance, not just that I went through, but Jay went through. And what this is doing is actually leveraging models that are translationally relevant. To end you with this and in closing, what this means is that we have an FDA-endorsed approach that relies heavily on the biocytogen transferrin receptor knock-in mouse. And it's allowed us to move forward for biodistribution tox species and the evaluation of first in human dose. They aligned that NHPs are irrelevant because they're pharmacologically irrelevant, and it allows us to think about how to run a rodent-only approach to regulatory approvals. That cross-species translation saves time, energy, money, and effort. And then on top of that, too, that surrogate capsid validation and that work means that you can re-leverage data that already exists. I'm really proud of the work that we do at Apertura. I think that we have a focus on what actually matters for the field. And critically, that's having collaborations. That's not just collaborations with other companies that are licensing our capsids, but collaborations with groups like Biocytogen that understand how to develop important critical tools for outcomes. We've been able to develop this approach. It's a preclinical and regulatory roadmap that has FDA endorsement. I'm excited to see others do this as well. We've been able to move forward in the field, and the only way we do that is through our collaborations with groups and with the team that we have. With that, thank you so much for your time and attention. I want to hand it back to Jay. Jay, I think you're on mute.
And while we're waiting for Jay to pull up his slides, feel free to go ahead and drop in the questions you have. We will have the Q&A session right after Jay's presentation.
So to continue with the previous slides, I have many thanks to Andrew for their inspiring story. In addition to the example set by Apertura team, this page also shows an exciting summary about Biocytogen's drug target humanized mice, the achievements and why to choose Biocytogen's drug target humanized models. Our drug target humanized mice are widely used by pharmaceutical companies, especially for FDA and NMPA. And for the IND applications. Many drug candidates that use our drug target humanized mice in their toxicology studies received IND approval from FDA or NMPA or even both. More excitingly, 2 drugs received the BLA approval from NMPA. These approvals cover diverse targets and drug types, including monoclonal antibodies, bispecific antibodies, and ADCs. So I will showcase 2 targets, L4 alpha receptor alpha, and CCR8 in the next few slides. LQ036 is a novel inhalable nanobody for asthma that shows high affinity and specificity for human L4 receptor alpha. So L4 receptor alpha is in the central pathway that drives the airway inflammation, and also it is a clinically validated target with effective anti-L4 receptor alpha monoclonal antibodies. So L4 receptor alpha is highly expressed in airway epithelium, smooth muscles, and immune cells. Inhalable L4 receptor alpha biologics may offer superior local efficacy and safety. So the table on the right side shows the details of the GLP compliant repeat dose toxicology study result. So given the limited time, the key findings for this drug candidate, the toxicity is summarized on the left. So the no observed adverse effect level identified for this candidate is high as 20.9 milligram per kilogram, and there's no significant toxicological changes were observed across various measured parameters. The key cytokines and immune markers remain normal. The candidate shows low immunogenicity. Anti-drug antibodies were detected in only a few mice. The candidate is currently in phase 2A clinical trials in China. I believe you all remember that to be considered as a relevant species, the drug candidate needs to be pharmacologically active in these mice. So indeed, the drug candidate shows a dose-dependent efficacy in a series of assays in this slide and in the next slides. So the two slides shows the results from the efficacy study. Another successful case in the clinical trial is an anti-CCR8 monoclonal antibody. Although we're not able to disclose the client and drug name, the preclinical efficacy and toxicology studies were performed in MC38 tumor models using humanized CCR8 mice from Biocellogen. The results showed a decrease of tumor infiltrating Tregs population and the increase of CD8 positive T cells. Also, the candidate is well tolerated at therapeutic doses with no major safety signals. The milestone of this candidate received due IND approval and is currently in phase 2 clinical trials across various solid tumor indications. Toxicology data from drug-target humanized mice showing good clinical relevance later on has been published in many literatures and reports. So GMA161 is a good example. This GMA161 is a humanized anti-human CD16 antibody that does not bind to common Neckermann primates. And the GMA161 tested in CD16 humanized mouse models exhibits clinical relevance, relevant pharmacological and toxicological responses such as platelet recovery, neutrophil decrease, and target-mediated drug disposition. The repeat dose toxicity of this candidate in CD16 humanized mice also correlates to clinical observations. This long list of IND approvals is a good testimonial for our strong animal support and IND-ready support for IND application. So on our animal support side, we have over 10 years of gene editing expertise. So the animals we generated closely mimic the expression in human or wild-type mice. We also provide very comprehensive model validation data, including in vivo expression, functionality, drug binding, to ensure clinical relevance and reproducibility. We also have the large animal variant to provide rapid and synchronized animal supply. And for IND-ready support side, we have been working closely with major safety assessment centers to ensure the provision of all required materials, and we can provide high standard product data that can be directly used as supplementary files for IND application. We also have a very comprehensive production and breeding statistics that offers essential background for safety assessment centers to conduct long-term and reproductive toxicity studies. Biocellogen offers a diverse pool of models to contribute the new drug toxicology studies with established advantages in efficacy study and toxicology studies. We can cover different disease fields and covering different modalities. So in the next few slides, I will just quickly show some selected lists of our diverse models categorized by disease areas and modalities. The established advantage, as I mentioned earlier in earlier slides, the drug-targeted humanized mice can provide direct evaluation of the drug candidates for clinical trials. We don't need surrogates. And also the mouse strains, they preserve the human target specificity and the MOA. These animals are especially useful for antibodies with no cross-reactivity with non-human primates. This using the drug-targeted humanized mice also brings in additional advantages like cost saving, shorter timeline, and less test articles consumed. And this slide highlights that we provide thousands of strains covering antibody discovery, molecular antibody discovery, bispecific antibody discovery, ADC conjugates, CAR T cell therapy, nucleic acid drugs. All these catalog strains, they can be used for efficacy study, PK/PD study, and in today's topic, they are also useful tools for toxicology studies. And I will quickly go through a few lists. And these are popular targets in the inflammation and autoimmune disease field. We have humanized all these targets in our catalog. We also have a long list of useful strains that is covering the immuno-oncology field. So feel free to check out our catalog, and we have a long list. In ADC field, we can provide very comprehensive characterization of the strain, and this on the right side is a very short list. And we also have developed a long list of strains for cell engagers, like T cell engagers, NK cell engagers. For each our catalog strains, we prepare comprehensive data package. In the basic data package, we have the mouse status, growth curve, blood routine, and immune cell development, our major issues or organs, observation or reproduction statistics. But when you plan to use these animals for toxicology studies, we also provide further validation data for the verification of species correlation, such as target expression, tissues profiling, and functional verification, test article binding verification, test article activity verification, and yeah. All of these animal supply and the studies cannot be successful without a good vivarium and a breeding team. And we have very comprehensive breeding support for colony maintenance, for embryology and surgery, for quarantine, rederivation, genetic analysis, phenotypic evaluation, and breeding management. We can supply large amount of animals with the same age and with the minimum weight differences. Our breeding vivarium are all A-like certified, and we can provide over 800K mice per year. With all our shipments, all our animals we offer, we can provide comprehensive pathogen test to get approval from your ovarian. So at the last few slides, I will briefly mention that as a CRO, we provide preclinical pharmacology capabilities. The topic today is about toxicology studies, but we can also do in vivo efficacy studies, PK/PD studies, in vitro ex vivo studies covering different disease models, tumor models or disease models. So for all our pharmacology services performed at our Beijing site or Boston site, we have our client first policy. We provide money back guarantee, and will also cover the complementary overage animal cost. So I will just thank you very much for your attention. I will just stop here.
Thanks, Jay, and thanks, Andrew, for the great talk. Very insightful and informative. I think we've already got quite a few questions in the Q&A box, and I saw that Andrew has already answered a few there. But just to bring those answers to the whole audience, let's still try to go through them live and take a look together now. And to our audience, if you haven't submitted your questions yet, feel free to do it right now. Okay, I think the first question here, and I guess most of the audience will be interested in, is were there any strategies that really helped during your early conversations with FDA?
Yeah, so I appreciate that. I tried to do my best to answer some of these questions that were sent in directly for folks. Please feel free to reach out to me if we don't have time. Honestly, the most helpful thing you can do is stop and read. Almost all the information that you need is publicly available online. The FDA guidance has been there. It's been clear for a very long time. There are only 7 questions to answer in order to move something into clinical trials. And outlining your strategy, why it makes sense, is really the name of the game there. So I think the most helpful thing we did was stop, read, and then define what we were trying to do and why it was either something that had already been done or already been considered, or if it was completely new.
Awesome. Thanks, Andrew. And next question here. For drug candidates that do cross-react with non-human primates and show good tissue distribution, is it possible to use humanized mice for tox?
Maybe I can answer a little bit of this and then I'll throw it over to Jay, because we talked about this a little bit during our prep call. So it's been my experience that there are a lot of things that can cross-react, but cross-reaction does not mean that's translationally relevant in this level of cross-reaction. So critically, there are certain receptors that if you engage them a little bit, do some things, but if you engage them much more, they do other things. So as a result, if you have something that does cross-react, but is not informative, or you can demonstrate or explain why it's not informative, in that case, you might be able to transition solely to a humanized model. Now, in my perspective, I think that's always better because our goal is to go into people, which are models of humans, and have an easier path there that's more translationally relevant. But Jay, I don't know, with your experience, how do you think about that?
Based on our experience and past projects, we also noticed that the cross-reactive candidates, we can also use drug-like humanized mice for the toxicology studies after a thorough discussion with the regulatory bodies.
Okay. Thanks, Jay, and thanks, Andrew. Let's go to the open questions because there are more coming up. The first question here, how many humanized mice are typically needed for a formal tox study?
It's a really good question. The true answer to that is it depends. And the thing it depends on is the modality that you're using. So if it's a small molecule, if it's a large molecule, if it's a one-time or if it's a repeat, as well as how rare the disease is and what the goals are of that tox study. So for certain things that are extraordinarily rare or N of 1, you can imagine that you're going to have a much smaller tox study than what you would have for a drug that's going to be used for a very, very wide population. Also noting that the dose and the dose regimen, so if it's one pill or if it's ascending dose, does change those things. So that's a specific question that you're going to need to speak with the FDA about to get a formal set of feedback. But there is no requirement in guidance that says you must have a statistical powering of your efforts. That's helpful, but is not necessarily required.
Awesome, great to know. And next question here, does Biocelgen provide both non-GLP and GLP tox service? I guess this one goes to Jay.
We currently provide non-GLP toxicology studies. Yeah, GLP work will go through those assessment center. So many of our clients, they purchase our animals and then they ask us to ship the animals to these centers for the GLP work.
Thanks, Jay. Yeah, go ahead.
Sorry, yeah, I would just want to emphasize, we work closely with these centers on the workflow, on the documentation, on paper. So the communication is very smooth.
Mm-hmm. I can attest to that.
Thank you very much.
Thanks, Jay. Thanks, Andrew. Next question here. How to integrate humanized model with disease model to show the on-target efficacy, considering that these two models generally separate or need to build a humanized disease model?
I can try to answer that. I mean, in the AAV case, I outlined how to do that. And one of the ways to do that is using a surrogate approach. where you're looking for that. Now, if you're looking at other modalities, be that a small molecule, an antibody, an ASO, or others, that might become more complicated. I'll note there are multiple products which are approved, which did not have any ADME PK engagement because there's no appropriate animal model. So in some cases, it's actually cell-based work only that can drive that forward. So the answer to your question is, it depends, but I tried to give you a couple of answers of what it depends on and not just give you the consulting answer.
Yeah, we can use our genetically modified drug-targeted humanized mice to induce disease models, and then we perform efficacy study and toxicology studies in these induced disease models.
Great. Thanks, Andrew. Thanks, Jay. Next question here. In generating a humanized mouse model for, say, a receptor which is not conserved among species, how do you ensure that the downstream signaling pathway will be the same in mouse and human?
I think there's an overall part of this of like, it's not necessarily the case that you're going to be able to. There's no such thing as perfect, but I imagine that working with groups like Biocytogen that have a lot of experience here can help you think about how to characterize those animals after you develop that model. I would say I would be pretty surprised if you have a receptor that's not conserved across species at all. There might be, it's usually the case where you have multiple copies of something rather than something not there at all, but I don't know, Jay. But I think the answer there is you have to do the science, right?
Yeah, and most of our drug-targeted humanized mice were in situ replacement. So we replaced the mouse sequence with human sequence. And the first validation we want to confirm is to compare these drug-targeted humanized mice with wild-type mice to see if we observe obvious differences. Yeah, and of course, over time, we'll accumulate more data, functional data, or expression profiling data. We can compare that with human as well.
Awesome. Let's go to the next question here. Similar to a previous one. So in the event NHPs are still relevant, can you justify only using transgenic mice for preclinical studies or tox studies, or would the FDA or EMA still require NHP studies?
The formal answer to that is it depends on what your modality is and what the purpose is. So the critical thing that you're looking at, you want to answer is why is it relevant? So if something is relevant, there is an idea of the 3 R's principle, which is reducing, refining, and replacing. Reducing means reducing the number of animals in a study. Refining means doing 2 studies at once, so a tox and distribution study together rather than a tox study and a distribution study. And then replacing means actually going lower down in the animal complexity, so we're not using as many non-human primates. That's an FDA policy. So noting that there's no requirement that's hard and fast, it's going to be about a conversation here. So if you have an opportunity to define and demonstrate that the transgenic mice are relevant and are more useful, you're going to be getting higher N and better data off of that, and that usually will go far. But if the route of administration is different or complex and is not relevant in the mouse, then you're probably not going to be able to. I would suggest having a conversation outlining the pros and cons and building a table like I showed.
Awesome. Thanks, Andrew. Okay, the next one here. Also curious, after enhancing brain targeting across the blood-brain barrier compared to IV injection, how is liver targeting affected? Does it remain similar or is it reduced? Additionally, what is your strategy to study off-target effects and to translate or bridge the delivery patterns observed in humanized mouse models to clinical settings?
So I'm pretty sure this is an AV question that was asked because I saw the previous one. We have published data on the first generation of the capsid. That's actually, I think, the first publication ever that mentioned the Biocytogen mouse. It's in Science from Ben Deverman's group with our first generation capsid showing a full and complete biodistribution in that animal. The capsid that I was talking about, TFR1 CapX, is an improved version of that. We have full distribution data on that. Happy to talk. I think we have presentations that are public that you can see as well. We see a reduction in the amount that goes to the liver, which is great. It doesn't spare the liver though, and I think it's actually an important thing to hit the liver if you're looking for neurometabolic and not just neuro. From a perspective of off-target effects and translating and bridging delivery patterns seen in the mouse versus other settings, you have to actually run those clinical studies to do it. And frankly, clinical studies do not give you distribution data. The only time they give you distribution data are in the unfortunate events where there's a death. So to that end, you'd be looking at target engagement data, not looking at distribution data. That bridging of those efforts and patterns has been demonstrated not just in AAV's case, but in many other molecules where IV routes of administration for BBB crossing things are relevant in mice as well as higher species. So it's not a no, but rather I think it's already there for all of that data.
Great, thanks, Andrew.
Mm-hmm.
Okay, up to the next question. Are the humanized mice appropriate models for pharmacokinetic studies, and what special considerations should we keep in mind?
That's a pretty broad question, Jay. Do you want to try taking that, or I can help out on it too?
Yes, please. Please help first.
Sure. I mean, it depends on what receptor you're talking about. So pharmacokinetic studies, if you're doing a small molecule, are going to be very different than if you're doing a large molecule, which is going to be very different than if you're doing a cell and gene therapy. So I think there's part of this that depends on what is your modality. Receptor binding actually might be more relevant when you're doing a pharmacokinetic study if you have a humanized receptor. Now, then there's a question, though, about like other protein binding and whether or not that's changed because say serum albumin or other things that you're coming to. Again, this does come to considerations of what modality you're using, what the dosing frequency that you're using is, et cetera. Now with that though, there's a great body of data out there already about how to approach this when you don't have really cool models like what Biocytogen has put together. And I think all of that information is still going to hold true and relevant. I don't know, Jay, I mean, I always think of it as like it's modality first for answering any of those questions.
Yeah, I totally agree with you. Yeah, actually, PK studies is a major part of our pharmacology projects. We conduct a lot of PK studies using our catalog strains, and of note, the catalog strains they may have multiple targets humanized. That's contribute to the distribution of the test articles. Yeah.
Great. Thanks, Andrew. Thanks, Jay. I think we're running a bit short on time, so let's take one last question here. Let's see. What is your typical timeline to generate the knockout mouse model?
That could be quite short, 3, 5, 6 months. It depends on the exact targeting strategy. Yeah.
Okay. Awesome. Unfortunately, I think that's all the time we have for questions today. So just to wrap, yeah.
Sorry, can we take one more question?
Oh yeah, of course. Which one do you have? One you want to take?
The historical control data for transgenic animal models that you offer.
Okay, so I will just repeat the question. Do you have historical control data for the transgenic animal models that you offer?
Are we talking about the breeding statistics or what part of the historical part? So we keep close monitoring of our generations of the strains when we establish colonies. So for all the strains used for toxicology studies, we can provide the full history of the breeding history.
And in my experience, the Biocytogen's also done a lot of good work in generating baseline data about histology and other things for those animals so that you have a means of comparison. So you're not just comparing against the background strain, but actually that transgenic animal with all the experience they have in their hand. They do a good job of all that record keeping.
Yes, for thousands of the strains, we cannot provide comprehensive validation all at the beginning, but over time, we always accumulate more data for as reference in each strain.
Great. Just to make sure there's no new questions coming up. All right. I think that's all we have for the question today. Okay, just to wrap up, first of all, a big thank you to Andrew and Jay for sharing your insights, and thank you to everyone in the audience for the great questions. If you have any follow-up questions or you want to learn more about, tox studies or humanized mice, feel free to reach out through our website at biocytogen.com or connect with us on LinkedIn. We will be happy to continue the conversation. And if you missed any part of today's session, this webinar will be available on demand. You can request a recording on our website as well. Lastly, before we let you go, just want to give you a quick look at what's coming next. So Biocytogen has more webinars on the way that will explore toxicology topics in more detail. We will be covering everything from, pre-IND studies like immunotoxicity, on-target off-tumor toxicity, and genotoxicity, as well as post-IND areas like reproductive and developmental toxicity, carcinogenicity, skin toxicity, and more. So stay tuned. We've got lots more to share in the coming months. All right, I think that's all from us for today. Thanks again for joining everyone, and we hope to see you at our next webinar. Thank you.
Thank you, everyone.